Welcome. This video will show you how to use the Multi-Locus Sequence Typing MLST app. MLST is an ambiguous approach of characterizing isolates of bacterial species using the sequences of internal fragments of housekeeping genes. It identified internal nucleotide sequences of approximately 400 to 500 base pairs in multiple housekeeping genes. Unique sequences are assigned an integer number, and a unique combination of alleles at each locus specifies the sequence type. Let's start. The MLST feature can be found in the Genome Analysis module after being downloaded. The selection of MLST option will prompt you to this wizard to provide some information about the input files. First, select the type of input files. This app allows raw reads as well as assembled files or context. Then, click in the Add File button to select all the files to be analyzed. For FASTQ files, establish the pattern to recognize upstream and downstream FASTQ files. Lastly, establish the MLST configuration by selecting the corresponding organism name. In this example, we're selecting Flavobacterium psychrophyllum. And then click Run. Let's talk about the results. When the MLST analysis has finished, click OK in the notification box and take a look at the MLST Results table. The first column will show the tags that contains a quick overview of the MLST results of each sample. It will generate 3 possible reports. Match, when the average identity and average coverage between all the housekeeping genes, templates, and the query reads or sequences is 100%. Partial, when the average identity and average coverage between all the housekeeping genes, templates, and the query is less or equal to 99%. Or no match, when the query sequence did not match any housekeeping gene template within the chosen MLST configuration. The next column is the name, which displays the input file name. Then is the sequence type column, which contains the corresponding MLST. Please note that for all partial results, the sequence type will have a number and an asterisk, which indicates that the sequence type number is not a 100% match. Next, you will find the average identity and average coverage columns. In these columns, you will find the average of the percentages identity or coverage of the alignment of all query reads or sequences that match a housekeeping gene template sequence in the database. Lastly, there is the notes column, which contains important information relevant to the sequence type results generated. Match results will not have any information in this column. However, partial results will display all the alleles with discrepancies. This discrepancy may indicate that a novel allele was found, errors, or SNPs. An MLST report will be generated. This report contains information relevant to the MLST run, including the input data, MLST configuration used, MLST results, and the parameters used for the analysis. Each sample or file will generate an MLST results and an MLST alignments report. This can be found by right-clicking on each file name. The report shows inputs and configuration information, as well as the MLST results for each allele with their respective identity and coverage percentages. The MLST report table will indicate what alleles match 100% and which alleles do not. Additionally, each sample or file will generate an MLST alignments report with a detailed alignment of each allele in the database and the corresponding match in the query sequence. Discrepancies between the query and database sequences are highlighted in red. This video shows the definition and application of the Multi-Locus Sequencing Typing, or MLST, how to run the MLST app from the OmicsBox fast and easy, as well as how to obtain and interpret the MLST results from the OmicsBox. If you have any questions, please do not hesitate in contacting us. Thanks for watching.